Okay, so next up is a company by the name of Naloomar. Just a quick blurb on them. You know, there's so much information that can be gleaned from our DNA. And, you know, our DNA can actually act as barcodes that can unlock health vitals and basically help in creating new treatment opportunities in the field of health. So to learn more about what I'm alluding to is Naloomar, led by Dr. Andy Fraser.
Thank you very much, Hassan. Just checking that you guys can all see my slides fine. And so, well, thank you very much all for being here on this wintry morning. I'm Andy Fraser, a prof at U of T, and I'll tell you about the new tech we've developed at Naloomar, which we think is going to transform how we see small molecules in multiple different sectors of healthcare. So what do I mean by small molecules, and why should you care about them? So right now, you have a complicated mixture of thousands of different small molecules running around your bloodstream: glucose, vitamins, cholesterol, hormones. If we could measure those accurately on a regular basis, we'd have a beautiful, accurate vision of your health and how it's changing over time. And so the diagnostics community clearly needs to measure these in an efficient way. On the flip side, in the research community, the way we go in and treat people with healthcare problems is with small molecules, drugs and various metabolites and natural products that you can use as additives. So they also need to be able to track these, produce these, and measure their output. So how do we currently measure small molecules? Right now, we use a machine called a mass spectrometer, and that's been the case for 60 to 70 years. It's an extremely powerful machine, and you can throw almost any kind of small molecule at it, and you will see a beautiful peak on this. So if this already works, what's the problem with the mass spec? And the answer is it's a major bottleneck right the way across healthcare. A mass spec costs about $1 million. It's the size of a car, and you can only use it if you're a trained tech in a diagnostics lab. You will never use a mass spec in your home. You will never make a measurement on a mass spec. So that's a problem for the diagnostics community. For the research community, the problems are completely different. They need to be able to measure tens to hundreds of thousands of samples a day, and the throughput of an individual mass spec is about 1,000 a day. And the other major problem is the sensitivity. We want to be able to measure small molecules down to the scale of single cells, and mass specs simply cannot go there because there's not enough of these molecules in a single cell. So if we're not going to use a mass spec to measure these, what's the alternative? And that's where we come in. We've developed a technology that can satisfy both communities. We can use our sensor technology both in simple point-of-care devices for home diagnostics and deal with the problems of cutting-edge research where they need to be doing hundreds of thousands of millions of measurements in parallel, and we can go down to the scale of single cells. So what is this magic and how does it work? Simply, we get away from the problem of looking at small molecules by transforming the whole problem into just reading out DNA sequences. And the way we do this is we pair up each different small molecule with a unique short DNA tag, which we call barcodes. So now instead of looking at these complicated shapes of small molecules, all we have to do is use the awesome power of DNA sequencing technology to read out tags, and that lets us count and measure the levels of any small molecule we want in a complex setting. But how exactly does that work, right? You have a mixture of thousands of different small molecules. How do we go in there and physically add those barcodes so that we can count them? And that's the core of the technology that we've developed. What we use is sensors that we develop here at Naloomar, and they essentially work like this. Each sensor recognizes and detects one and only one small molecule. So there's one sensor for glucose, one for cholesterol, and so on. And each sensor is physically attached to a unique barcode. So when the target comes in, the sensor sees its target and releases its barcode. And now all we have to do is grab and sequence those released barcodes, and we can measure each and every molecule multiplexed in parallel in an extremely complicated setting, because each of those barcodes is unique and specifies a single molecule that has been detected. Let me show you this working in action. So I'm showing you a range of different sensors that we've developed, shown here. So there's an antibiotic, ampicillin, an anti-malarial drug, piperaquine, THC, the active component of cannabis. We've got an amino acid, we've got cortisol, the stress hormone that's probably running through my system right now, and glucose. And in each case, what you see is absolutely beautiful. You titrate in the levels of the target, and you get this beautiful dose response coming up. So we can measure not just presence or absence of the small molecule, but we can count each sensor releasing each barcode as that concentration increases. And remember that because each sensor releases a different barcode, we can measure all of those things together in a single volume. If you look at comparable technologies, I talked already about mass spec. The key advantage that we have over mass spec is the cost effectiveness, obviously, because mass spec is extremely expensive in terms of running these in diagnostics labs and the instrumentation itself. But also it lets us reach areas that it really can't. We can multiplex massively to give us much greater throughput. And because what we release is DNA barcodes, and that it's trivial to amplify DNA millions of fold within an hour, we can reach right the way down to single cell, and that's critical for doing things like looking at drug uptake in solid tumors and so on. The other comparable technology would be antibodies. For me, would be the sort of most interesting one. And antibodies are tremendous reagents, but they have a key blind spot. They're really bad at detecting small molecules, whereas they're great at detecting proteins. And we fill that exact blind spot with our Naloomar technology. We will see small molecules, and antibodies will see peptides and proteins. And that's a beautiful sort of mix of technologies, and we're providing that other side. The markets that we think that we're going to be able to address within the research space are single cell detection. So currently, you cannot look at small molecules in single cells because mass spec is not sensitive enough. There is no technology that lets you do that. So that's pure blue ocean. That market is already $3 billion a year market, and it's growing rapidly, as you can see. The other area that we're extremely excited to go after is synthetic biology, and that's the bioindustry of the future. Rather than making drugs and small molecules in bioreactors and chemical factories, what bioindustry is turning towards is getting cells to make those small molecules for them. Cells are phenomenal organic chemists, and what synthetic biology needs to do is measure the level of small molecules in hundreds of thousands of cells so they can identify which ones are producing them the best. And our sensors work hand in glove with synthetic biology, and I'll come back to that in a couple of slides. As you can see, that's already almost a $10 billion market and growing extremely rapidly, and that really is the wave of the future. Within the diagnostic setting, the nearest comparison that we have would be you can go home and do a comprehensive metabolic panel, and that looks at about 14 different things, and if you're lucky, you get to do one of those once a year. We think we can expand that panel at least tenfold very rapidly, and that'll all be home diagnostics measurable on simple diagnostic devices that you can all use at home. Already a big market, and again, growing robustly. So I've told you these sensors work absolutely beautifully, and there's major need for small molecule detection. So given that, why aren't we doing this already? Why can't you already use these things, and why are we still using mass specs? And the answer very simply is there's very few sensors available. There's fewer than 20 available in the world, but you can see that they work absolutely beautifully. So our goal over the next 18 months is to get 1,000 sensors. That will establish us as being the single largest small molecule detection company, and it will allow us to take these sensors and put them on everything from single cell platforms to diagnostics devices. The business plan, quite simply, is that we will sell these sensors. The beautiful thing is they're extremely high added value. Producing any single sensor is a couple of $100, and that's enough for many, many tens of thousands of different assays. And that's the model of a company, other enabling technology like Illumina, where the vast majority of their profits comes not from machines to sequence DNA, but from the reagents that let you do that, and it's huge high added value. The types of sensors that we think we're going to be selling come in two completely different flavors. One is bespoke sensors. So, for example, a synthetic biology company comes to us and says, we need sensors to allow us to detect 100,000 assays of this very specific small molecule. We will build those sensors for them in partnership and deliver them to them. We're already doing that for a well-established synthetic biology company, and we're in discussions with a second one to do that. So we're developing 20 sensors right now, and that should be delivered at the end of the first quarter of 2022. The other route is that we are going to be building up our 1,000 sensor collection, and those sensors can be put either on diagnostic devices or straight onto a single cell platform And you're going to be able to measure every single one of those metabolites, so 1,000 different ones in single cells in multiplex. And that's where we're heading. So it's the sensors that we're going to be selling. That's the critical thing. In terms of timelines, we basically see the first stage through to Series A is building out into the research community because they have the most urgent need. That's building the partnerships further that we already have with synthetic biology specifically, and building our 1,000 sensors. And from Series A to Series B, we will be taking those and putting them onto platforms, both within the research community and single cells and the diagnostic communities. The IP strategy, our core tech is already filed. That's this multiplexable aptamer-based ligand detection. But as we walk through this timeline, we're going to have measurable sort of milestones of IP that we're going to produce. The 1,000 sensors themselves, of course, will protect, as well as the single cell platform. But we also have the diagnostics platforms. And specifically, I'm very interested in essentially taking a thing like your COVID rapid test or a pregnancy test, which says yes or no for a single thing. And we are able to put tens of things onto those same very simple devices, and instead of yes, no, make them entirely quantitative. And that's what we could imagine our first devices flowing out to the community in that way. What we're looking for is we're looking for a 2 million seed to take us through to Series A. That will let us grow these partnerships and set multiple more of them, especially the synthetic biology community, and also build our collection of 1,000 sensors. That money would be for salaries for technicians to produce the sensors, lab space and equipment, and for largely the sequencing costs to identify those sensors and check that they work in exactly the dynamic range that we want, as well as IP costs. But we note that we're going to defray some of that money with partnerships which are already ongoing, and with non-dilutable funding from grants like GAP, Genome Canada, and NSERC I2I. We already have one of those. We've just applied for the next tranche of that. In terms of the team of people we have already, our CEO is Maria Mercado, a PhD student with 5 years of genomics and metabolism experience. The key for Maria, she has extremely strong links to both the Toronto and Canada-wide life sciences community, which includes fundraising, advising new startups, and leadership roles, including setting up entire international conferences. June is the magician that's produced all the data that has driven our sensor technology so far, and she has an extremely strong background in analyzing the massive sequence data sets that we're going to get back from our barcodes. And I'm a prof here at U of T, but I have a very strong history in developing new platforms and technologies, both in the academic sector, but also in the private sector. I co-founded a startup previously, Camina Biosciences, which exploited my new inventions in DNA synthesis technology. That's ongoing in the UK, and I still have equity there, but I've stepped back from all the science so I can focus on this. And in addition, we're extremely lucky to have recruited Stuart Milstein and Ben Lehner onto our scientific advisory board. Stuart Milstein's speciality is developing high throughput platforms to look at biology, which is exactly what we're doing. He's currently at Coro Bio, but he was previously at Alnylam, where he identified the first RNAi therapeutic. And Ben Lehner is a superstar of European science, winner of the EMBO gold medal, and he's a world leader in looking at massive sequence data sets and drawing out patterns and meaning within them, which is exactly where we're going to be with our barcodes. So we're extremely excited to get these guys on board. So I hope what you got from this is we have a beautiful new technology that for the first time in 60 to 70 years will let us look at small molecules with new eyes outside of the mass spec, and it should drive and enable massive progress both in the fields of diagnostics and research, and especially exciting is going to be the synthetic biology, where we're already making progress. So I'll take questions. If you want to talk to me afterwards, I'll be in the session room, and at any point, please feel free just to email us at info@naloomar.com, and we can talk to you anytime in the future. All right? Thank you very much, everyone.
Thank you, Andy. That was an awesome presentation. We got a couple of questions with about 7 minutes. First question from Mark. Are you planning to use AI or ML in your sensor design?
Yeah, so this is one of the funniest things with the type of sensors that we make. They've been around for 15, 20 years. And so far, there has been no progress whatsoever from using AI and machine learning for these. And that's kind of a mystery, because if you look at it right now, Google or AlphaFold can look at a protein structure, and they can fold up that protein de novo and predict what the structure is going to be with incredible accuracy. So why can't we do that with something as simple as small molecules? And the answer is there are no training sets. Like, as simple as that. The way people would have made these sensors in the past is they take an unknown billionth of a billionth of 1% of the possible sequences. They have no idea what they started with, and they go through an extremely complicated process to get to one sensor. What we do, because we can measure every single sensor, and we can look at these, we search for these 10 million in parallel, because our barcodes let us count every single one. We know every single one of 10 million sensors quantitatively how it saw each of our targets. So the reason we're going for the 1,000 sensors is once we have those data, we'll have had 10 million sensors quantitatively against 1,000 sensors. At that point, honestly, that's a trivial machine learning problem. So the next phase from Series A to Series B, I suspect most of our sensors, we won't have to use anything in the lab at all. It'll be algorithms that'll predict them. That'll be empowering.
Great. Next question. What is the current throughput? 12, 24, 96 wells? How does it grow over time?
Right. So 96 wells is where we're going. The key change that we have made, or at least one of the key changes that we're making, is we have greatly limited the sequence space that we need to look at. So typically, people look at the possible number of sequences they're looking at to look at a sensor is greater than the number of stars in the universe. So not a very tractable problem. So they have to do many, many rounds of amplification and change. Because we can look at every single sensor in a single sequencing run, we don't need to go through rounds and rounds and rounds. We just look all of our sensors, a single assay, and we read out what comes out and what binds. And we can do that 96 at a time and just walk through it. So that is how we get to our 1,000 rapidly. And that's a major change. And so, yeah, the throughput is extremely good.
Great. Next question. How will you address regulatory considerations?
So that's clearly an issue within the diagnostic space. That is why initially we are going between now and Series A after the research space, which is completely different, but we don't have to worry about those. And so at the point that we have our 1,000 sensors and we know all the responses, we will know which ones of those are diagnostically valuable. And that's where we'll end up by putting all the effort into the regulatory stuff. But that comes substantially after Series A. And the reason, honestly, for that is we know right now there's this desperate need within the research community for sensors. The diagnostic space, it will take people longer to accept that these are the way to do home testing. And that's why we've shunted our entire business plan towards research for the sort of initial 18 months, sort of 2-year timeframe.
Okay. Next question. Can you tell us a bit about the research you did to support the idea that your technique has the resolution to accurately distinguish thousands of similar small molecules?
Okay, so that's essentially a specificity question, if you like. So the question is, okay, so those graphs that I showed you right there, those were in buffer, one sensor, one small molecule being titrated in. But I could have shown you exactly the same curves, and I'm happy to talk about the session room. I can walk you through the data. We can take the lysate of entire cells and do exactly the same experiment, and we get indistinguishable data. So we can see each one of those single molecules in the context of many, many thousand different ones with absolute specificity. And don't forget also that because we're looking at every single one of our 10 million sensors against 1,000 targets, and we will know quantitatively how everyone behaves, we will know specificity just straight from the data. But at least for the sensors that we already have, they're highly specific. That's the way we've been looking for them. Because if that didn't work, then the entire approach would founder.
Okay. Next question. What do you think will be the first application of your sensors in single cells?
In single cells, I think the most likely is going to be within solid tumors. So we already know back from Warburg, so almost 100 years ago, we know that the centers of tumors are extremely different metabolically from the outside. What we can't tell is how different that is. Is it only 2 flavors or is it many flavors? And the more data that's coming out, this week there was an entire new one where tumor cells are metabolizing completely differently than had been known before. But the other critical thing is drug uptake into single cells. When you treat a solid tumor with a new drug, many of the cells will not die, but you have no idea whether they got enough of the drug, whether they metabolized the drug, or whether they excreted the drug. Because we can look at single cell resolution, we can get a map not only of how they've responded at the genomic level, but we can also see exactly the uptake and metabolism of each drug and how they've impacted the cells. And that's game changing. We've already talked to DNA repair companies that are producing drugs, and we're going to talk to them more about making sense as for what they're looking for.
Awesome. And the last question, which customers are you considering in terms of going to market? Is it small pharma, big pharma? What is your strategy of adoption of your solution by targeted market?
So I think the area that we're going into hardest is synthetic biology, and we have been talking to everybody from new startup synthetic biology companies through to extremely well-established synthetic biology companies. They all have the same problem, which is right here sitting in the lab, I have 100,000 different yeast cultures. They're all producing different levels of what I need, these extremely high-value small molecules, and we're unable to detect them with throughput. They're needing to spend tens of millions of dollars on mass spec to make that plausible, and so that bottleneck from them is absolutely excruciating, and we can relieve that very rapidly, and that's exactly what we're doing already. So we're already partnered up under an NDA with one of these companies, and we're talking to the second, and I think that will grow rapidly, and that is really where we want to go as the sort of first market, because it's exploding as an area of biotech.
Awesome. Andy, thank you very much.
Thank you very much, everyone. I'll be in the session room, so if you have any other questions, please come and join.
Take care.